Venetoclax is a targeted cancer drug that works by inhibiting the BCL2 protein, which is important for cancer cell survival. This presentation will explore the problem of resistance to venetoclax that has emerged in cancer treatment, and discuss potential strategies to overcome this challenge. Understanding the mechanisms behind venetoclax resistance is crucial, as it will inform the development of more effective cancer therapies. We'll cover the latest research in this critical area of cancer treatment. The BCL2 protein family plays a critical role in regulating apoptosis, or programmed cell death. These proteins are divided into three main categories: anti-apoptotic proteins like BCL2 and MCL1 that prevent cell death, pro-apoptotic proteins like BAX and BAC that promote cell death, and regulatory BH3-only proteins that can activate the pro-apoptotic proteins. Anti-apoptotic BCL2 binds to the BH3-only proteins, preventing them from activating BAX and BAC and blocking the pro-apoptotic pathway. When the BH3-only proteins are able to bind to BAX and BAC, they promote the recruitment and oligomerization of these proteins to form pores in the outer mitochondrial membrane. This pore formation releases cytochrome C from the mitochondria, leading to proteolysis and ultimately apoptosis in cancer cells. Venetoclax is a targeted therapy that works by selectively binding to the BCL2 protein, which is overexpressed in many cancers. By binding to BCL2, venetoclax mimics the action of BH3 proteins, which are natural regulators of apoptosis. This displacement of BH3 proteins, like BIM, allows them to activate BAX and BAC, the key executioners of the apoptotic pathway. The activation of BAX and BAC leads to the formation of pores in the mitochondrial membrane, release of cytochrome C, and ultimately the induction of programmed cell death or apoptosis in cancer cells. This select targeting of the BCL2 protein is the key mechanism of action that makes venetoclax an effective treatment for certain types of blood cancers. Venetoclax is an important targeted therapy for several types of blood cancers, including chronic lymphocytic leukemia, Kia, acute myeloid leukemia, AML, and other hematological malignancies. In Kia and small lymphocytic lymphoma, SLL, venetoclax is approved as a monotherapy treatment option. For AML, venetoclax is approved in combination with other drugs as a treatment approach. Venetoclax also shows promise in treating other blood cancers like follicular lymphoma, mantle cell lymphoma, and Waldenstrom's macroglobulinemia. However, many patients experience relapse within months to years after initial remission, indicating the emergence of resistance to venetoclax in some cases. It's crucial to continue researching ways to overcome this resistance and improve long-term outcomes for patients with these challenging blood cancers. Increased expression of other anti-apoptotic proteins like MCL1 and BCLXL can drive resistance to venetoclax. MCL1 overexpression is common in venetoclax-resistant cells, especially in monocyte phenotype AML. MCL1 binds to released BIM, preventing apoptosis. BCLXL upregulation is a significant contributor to venetoclax resistance in Kia, as it has a higher affinity for BIM than MCL1. The PI3K, AKT, mTOR pathway regulates expression of anti-apoptotic proteins and can be activated by microenvironmental factors, leading to resistance. Strategies to overcome this include using MCL1 inhibitors like S63845, S64315, and VU 661013, as well as BCLXL antagonists like A1155463. MCL1 and BCLXL are important anti-apoptotic proteins that help cancer cells survive. Inhibiting these proteins can make cancer cells more sensitive to venetoclax, a BCL2 inhibitor. S63845 and S64315 target MCL1 and show synergistic effects with venetoclax in T-cell and DLBCL. VU 661013 and AZD 5991 are novel MCL1 inhibitors that can convert resistant leukemia cells to be sensitive to venetoclax. AMG 176 and AMG 397 are effective in recurrent mantle cell lymphoma when combined with venetoclax. A11556463 is a BCL-XL antagonist that can reverse resistance to venetoclax in multiple cancer types. These combination therapies targeting MCL1 and BCL-XL show promise for improving outcomes in difficult-to-treat blood cancers. The slide discusses resistance mechanisms to the cancer drug venetoclax, specifically focusing on BCL2 mutations. The first mutation highlighted is GLY101-VAL, found in 7 out of 15 chronic lymphocytic leukemia (KIA) patients who progressed on venetoclax. This mutation decreases venetoclax's affinity for BCL2 by 180-fold. The second mutation is Phe104-EL, discovered in relapsed, refractory follicular lymphoma. This reduces venetoclax's affinity by approximately 40-fold. Mutations in the ASP103 codon, resulting in tyrosine, glutamic acid, or valine residues, affect the hydrogen binding of venetoclax to BCL2. Most resistant patients harbor 2 or more BCL2 mutations, which severely impacts prognosis and treatment options. The slide discusses the structural impact of 2 key BCL2 mutations, GLY101-VAL and ASP103 mutations. The GLY101-VAL mutation is located in the BCL2 alpha-2 helix, where the substituted valine creates a stable extension of the helical structure. This structural change allows the helix to shift, reducing venetoclax binding while maintaining the ability to bind BH3 motifs in regulatory proteins. The ASP103 residue is crucial for hydrogen binding of the venetoclax drug to the BCL2 protein. Mutations at this position in the P4 pocket significantly impact drug binding. The ASP103 GLU mutation makes BCL2 more similar to BCL-XL protein, affecting venetoclax binding but not navitoclax sensitivity. The slide includes a detailed 3D rendering comparing the molecular structures of normal BCL2 protein versus the mutated versions. BAX is a critical protein that helps drive apoptosis or programmed cell death when activated. In AML cells, loss or mutation of the BAX gene can block venetoclax's ability to induce apoptosis. Over 20 different BAX mutations have been detected in patients resistant to venetoclax. Mutations in the C-terminal domain of BAX prevent it from properly anchoring to the mitochondrial membrane, which is essential for its pro-apoptotic function. Without functional BAX, venetoclax cannot effectively trigger the mitochondrial apoptosis pathway, leading to resistance. TP53 mutations are a key resistance mechanism that can emerge in patients treated with venetoclax. These mutations increase in frequency by over 2-fold in mantle cell lymphoma, MCL, patients after venetoclax treatment, becoming a major driver of disease progression. TP53 mutations or inactivation can decrease expression of BCL2, the target of venetoclax, reducing sensitivity to the drug in AML cell lines. TP53 mutant cells also show elevated MAPK pathway activity, leading to increased MCL1 expression which can compete with venetoclax binding. TP53 knockout cells exhibit an altered protein balance, with increased BCL-XL and decreased NOXA, PUMA, and BAC, inhibiting apoptosis and contributing to resistance. Understanding these TP53-mediated resistance mechanisms is critical for developing strategies to overcome venetoclax resistance in hematological malignancies. This slide covers some of the key genetic mutations that can contribute to resistance to venetoclax, a targeted cancer therapy. First, we have FLT3-ITD expansion, which is a major driver of resistance in acute myeloid leukemia, AML. FLT3-ITD causes ligand-independent activation of the receptor, leading to downstream signaling through PI3K, AKT, mTOR and MEK/ERK pathways. Next, we see BRAF mutations, which have been detected in chronic lymphocytic leukemia, KIA, patients with acquired resistance to venetoclax. These mutations affect important signaling pathways. Notch1 alterations are also associated with venetoclax resistance in KIA, as they influence cell survival pathways. Finally, homozygous deletions of CDKN2A, B, which impact cell cycle regulation, have been observed in patients resistant to venetoclax. Understanding these resistance mechanisms is crucial for developing strategies to overcome them and improve outcomes for patients. The slide presents a detailed diagram of the FLT3-ITD mutation and its effects on cellular signaling pathways. The FLT3-ITD mutation causes ligand-independent activation, triggering the PI3K, AKT, mTOR and STAT5 signaling cascades. This leads to changes in protein expression, increasing levels of MCL1 and decreasing BAD and BIM, ultimately inhibiting apoptosis. FLT3 inhibition by compounds like HQP1351 can synergize with venetoclax, blocking FLT3 phosphorylation and downstream signaling, down-regulating anti-apoptotic proteins. This combination therapy enhances apoptosis, as evidenced by increased caspase-3 and PARP cleavage in AML cells. Resistant cells have undergone a metabolic shift, moving from relying on amino acids to using fatty acids as their primary fuel source. This metabolic change leads to increased production of the coenzyme nicotinamide, which in turn boosts NAD+ levels. Higher NAD+ supports more efficient oxidative phosphorylation, OXPHOS, in the mitochondria, generating more ATP for the cell. The energy sensor AMPK is also activated, further promoting cellular respiration and ATP generation. Structural changes in the mitochondria, like tighter cristae formation, provide even more energy capacity for the resistant cells to survive. Leukemia stem cells, LSCs, typically rely on mitochondrial amino acid metabolism to survive. Venetoclax and azacitidine can kill LSCs by reducing amino acid uptake and catabolism, and inhibiting the mitochondrial electron transport chain. However, some resistant LSCs can shift to fatty acid metabolism instead, bypassing their need for amino acids. ROS mutations in resistant LSCs are linked to increased carnitine synthesis, fatty acid metabolism, mitochondrial fatty acid extension, and beta-oxidation of long-chain fatty acids. This allows the resistant LSCs to generate energy for survival despite venetoclax treatment. The metabolic adaptations in resistant LSCs represent an important challenge in treating certain types of leukemia. Resistant cells have amplified regions containing genes for the regulatory subunits of AMPK. AMPK activation promotes cellular respiration and ATP production, while limiting ATP-consuming processes. Activated AMPK leads to phosphorylation of the BAD protein by the PI3K, AKT pathway. Phosphorylated BAD binds to 1433 protein, preventing its mitochondrial translocation and blocking apoptosis. This AMPK-mediated signaling contributes to venetoclax resistance in these cells. Mitochondria are the powerhouses of cells, responsible for energy production. In resistant cancer cells, we see several key changes in mitochondrial function. Increased expression of the OPA1 protein, which helps maintain the structure of mitochondrial cristae. Higher maximum oxygen consumption rate, indicating enhanced metabolic activity. Elevated levels of reactive oxygen species, ROS, increased amino acid, coenzyme, and ATP metabolism for greater energy production. These mitochondrial adaptations help resistant cells survive the effects of the drug Venetoclax, which normally induces apoptosis by disrupting mitochondrial function. Understanding these mitochondrial changes is crucial for developing new strategies to overcome drug resistance in cancer. Mitochondrial protein synthesis is a key process that can contribute to drug resistance in certain cancers. We've identified several mitochondrial ribosomal proteins that play important roles in this resistance mechanism. MRPL54 is a component of the mitochondrial translation machinery that was identified in a CRISPR screen of resistant cells. MRPL17 regulates mitochondrial protein synthesis and can alter metabolism in resistant acute myeloid leukemia cells. RBFA is a ribosome binding factor that, when expressed abnormally, contributes to Venetoclax resistance through changes in mitochondrial function. Combining Venetoclax with the antibiotic tedazolide, which blocks mitochondrial protein synthesis, can help overcome this resistance mechanism. Venetoclax is a targeted therapy that has transformed the treatment of certain blood cancers, but resistance can develop over time. When this happens, we need to explore alternative treatment strategies to overcome the resistance. This slide outlines 4 key approaches we can consider. Targeted combinations personalizing treatment based on the specific resistance mechanisms at play. Anti-apoptotic protein inhibitors using drugs that target proteins like MCL1 and BCL-XL to restore sensitivity. Signaling pathway modulators leveraging inhibitors of pathways like BTK and PI3K, as well as agents like arsenic trioxide. Metabolic targeting exploring OXPHOS inhibitors and modulators of mitochondrial function. BTK inhibitors like ibrutinib can be an effective treatment option for patients with chronic lymphocytic leukemia, CLE, who have progressed after receiving Venetoclax. BTK inhibitors work by decreasing levels of the MCL1 protein while increasing the BIM protein, which enhances the dependence on BCL2 and increases the affinity for Venetoclax, creating a synergistic effect. Clinical studies have shown that BTK inhibitor therapy after Venetoclax failure can result in high response rates and durable remissions, with a median progression-free survival of 34 months and median overall survival of 42 months. Combination approaches using BTK inhibitors like ibrutinib plus Venetoclax have also shown promising efficacy and safety in relapsed, refractory mantle cell lymphoma, diffuse large B-cell lymphoma, follicular lymphoma, and acute myeloid leukemia, suggesting broad applicability across hematological malignancies. MCL1 is an important anti-apoptotic protein that can confer resistance to Venetoclax, a key therapy for hematological cancers. Inhibiting MCL1 can help overcome this resistance by freeing up the pro-apoptotic protein BIM, which can then induce cell death. Several novel MCL1 inhibitor compounds are in clinical development, including S63845, AZD5991, VU661013, and AMG 176. These MCL1 inhibitors have shown promising synergistic effects when combined with Venetoclax, leading to improved outcomes in various blood cancers. The slide highlights the molecular structures of these different MCL1 inhibitors, visualizing the drug discovery and development process. Arsenic trioxide is a promising agent for overcoming drug resistance in cancer cells. It works by modulating key signaling pathways that regulate the anti-apoptotic protein MCL1. Arsenic trioxide leads to phosphorylation of MCl1, which destabilizes the protein and triggers its degradation. This releases the pro-apoptotic protein BIM, which was previously sequestered by MCl1. The restoration of BIM activity then induces apoptosis in cells that were resistant to venetoclax and other therapies. Overall, arsenic trioxide shows potential as a resistance-reversing agent that can resensitize cancer cells to treatment. Anti-CD20 monoclonal antibodies like rituximab and obinutuzumab are important therapies for B-cell malignancies. Rituximab alters the expression of BCL2 family proteins, helping to overcome resistance to venetoclax-induced apoptosis in chronic lymphocytic leukemia, KIA, and other B-cell cancers. Obinutuzumab is a next-generation anti-CD20 antibody that works through similar mechanisms to enhance sensitivity to venetoclax. Combining venetoclax with anti-CD20 antibodies provides significant clinical benefit, allowing patients to overcome resistance by targeting complementary pathways. This slide introduces several metabolic targeting strategies that have shown promise in treating acute myeloid leukemia, AML. The first strategy is the combination of venetoclax and azacitidine, which restricts amino acid uptake, disrupts the TCA cycle, and inhibits the electron transport chain, leading to high response rates in AML clinical trials. The second strategy is 8-chloroadenosine, which interferes with fatty acid metabolism by down-regulating proteins involved in fatty acid oxidation, reducing OXPHOS levels to counteract resistance. The third strategy is a CYL-chlorohydroquinone, which produces OXPHOS uncoupling and sensitization to venetoclax in human leukemia cells. The fourth strategy is tigecycline, which increases cell sensitivity to venetoclax by inhibiting mitochondrial OXPHOS and affecting mitochondrial function. The combination of venetoclax and azacitidine has shown impressive results in clinical trials for AML patients. This regimen is highly effective, with significantly higher overall response rates, complete remission, and median survival compared to venetoclax alone. Importantly, the combination also leads to a higher rate of leukemia stem cell, LSC, elimination, which is crucial for long-term disease control. The synergistic effect of this combination targets multiple metabolic pathways in leukemia cells, overcoming resistance mechanisms and improving patient outcomes. The slide presents key efficacy data in a clear, visual format to highlight the benefits of this combination therapy. Mitochondria are the powerhouses of cells, responsible for energy production through oxidative phosphorylation, OXPHOS. Targeting mitochondrial function can be an effective therapeutic approach for certain diseases. This slide introduces 4 mitochondria-targeted therapies and their mechanisms of action. Tedazolid blocks mitochondrial protein synthesis, synergizes with venetoclax. 8-CL-ADU interferes with fatty acid metabolism, reduces OXPHOS levels. ACYL-chlorohydroquinone uncouples OXPHOS, sensitizes cells to venetoclax. Tigecycline inhibits mitochondrial protein synthesis, increases sensitivity to venetoclax. Non-Hodgkin's lymphoma is characterized by anti-apoptotic protein expression and clonal heterogeneity as primary resistance factors. In acute myeloid leukemia, abnormal OXPHOS and metabolic adaptations are widespread resistance mechanisms. Chronic lymphocytic leukemia often exhibits BCL2 mutations and OXPHOS abnormalities as common resistance pathways. TP53 alterations play a significant role in venetoclax resistance in mantle cell lymphoma. Personalized resistance profiling is key to overcoming venetoclax resistance. Comprehensive genetic and metabolic testing can identify specific resistance mechanisms in individual patients. This enables us to tailor treatment approaches to each patient's unique resistance profile. Exploring novel drug combinations that target multiple resistance pathways simultaneously is a promising strategy. Targeting anti-apoptotic proteins, signaling pathways, and metabolic vulnerabilities together can be more effective. Next-generation BCL2 inhibitors that can overcome common resistance mutations are needed. These new inhibitors must maintain efficacy even as the BCL2 protein structure is altered. Baseline genetic testing is crucial for identifying key mutations like TP53 and FLT3-ITD that can impact treatment response. Regular monitoring of minimal residual disease and early resistance markers during treatment allows us to detect resistance development early. At the first signs of resistance, we need to do comprehensive genetic testing to identify specific mutations in BCL2, BAX, and anti-apoptotic proteins. In advanced resistance cases, metabolic profiling to assess OXPHOS status and mitochondrial function can provide important insights. Closely monitoring for resistance development at each stage of the patient journey is essential to guide treatment decisions and optimize outcomes. Venetoclax is a targeted therapy that has revolutionized the treatment of certain blood cancers, but resistance can develop through various mechanisms. We'll discuss the key resistance mechanisms, how to diagnose them, and the treatment strategies to overcome them. Increased expression of MCL1 and BCL-XL can confer resistance. This can be detected through protein expression analysis, and treated with inhibitors targeting these proteins or BTK inhibitors. BCL2 mutations can also drive resistance, requiring alternative BH3 mimetic drugs or combination therapy. Loss or mutation of BAX leads to evasion of apoptosis, calling for therapies that induce cell death through other pathways. TP53 mutations render cells resistant to p53-dependent therapies, necessitating p53-independent approaches. FLT3-ITD expansion can co-occur, needing combination with FLT3 inhibitors. Metabolic rewiring, such as abnormal OXPHOS, can be targeted with OXPHOS inhibitors or azacitidine combinations. Venetoclax is a powerful targeted therapy for hematological cancers, but resistance can develop through various mechanisms. Key resistance mechanisms include upregulation of anti-apoptotic proteins, BCL2 mutations, loss of BAX, TP53 mutations, and abnormal mitochondrial function. Promising combination therapies that can overcome resistance include MCL1 inhibitors, BTK inhibitors, azacitidine, and mitochondria-targeted therapies. Monitoring for resistance is essential. This can be done through genetic testing, protein expression analysis, and metabolic profiling. Continued research into novel combinations and next-generation BCL2 inhibitors will further improve outcomes for patients with resistant disease.